embarrassing. You have no idea how embarrassed I was when I missed the half class. I was like, yeah, but don't you remember the 2 axes and the stuff? And I'm like, no, we don't. I'm like, really? I really didn't feel good. Very good. So how was the labs, quiz? Did you guys were you guys able to start a little bit of the presentation? And yeah, of course not. Kind of question. No, I'm kidding. Have you? No? So, little? It's fine. So we're gonna have the good thing is that we're gonna have enough time to, especially for this class, I tried to go to the first round, second round. The idea is really to get you guys going in the first round and try to get more or less the way in which some of you have been into 312. It's not like it changes. There's a lot of questions, I think, at the end, but it tends to go really well. The idea is really, up to this point, what I try to do, as I go, as I move through the years teaching, I am trying to talk less, which is very weird to me because I talk like too much. But the idea is really to have more conversations, during the presentation phase. So we go more like discussion, testing kind of thing. Okay. So for this next basically 2 weeks, we're gonna be going pretty much about this class of bacteria, viruses, fungi, and then the next class, which is pharmacological interventions and so on. So the idea here is that, from microbiology, obviously we're not gonna go, it is microbiology and so on, but we're gonna basically spend most of the time on bacteria and viruses. Okay? Are we gonna talk? Oops. We're gonna talk. Maybe you want more. Maybe you want. We're gonna talk a little bit on fungi and all the other stuff. And if we start going into like, are we gonna talk about like crazy toxins or prions or all these other crazy external agents? We're not. We may go into a case study here and there. Stop laughing, you guys, because now I'm honest. And the part is I'll try to avoid, because the idea is really not to go into so much detail into the others. But bacteria and viruses, they tend to be the focus of the class because they also tend to be the focus of the treatments. Okay? Viruses. It's not like it is the focus of the treatments itself, but it's almost everything you do for viruses is like supportive. Even if you take an anti-inflammatory for something that is caused by a virus, sometimes you take antibiotics because of secondary infections caused by virus, but actual treatment for viruses in domestic animals is zero. Okay, you either have prevention or you don't have much of a treatment. The other part is that if we want to talk about treatment of viruses, we'll have to go basically to cocktails used for HIV/AIDS, or basically serum positive for HIV in humans. Even aside from that in humans, it's also not common at all. Those are drugs that are, for the most part, lots of the viral infections that we have, they're temporary. They are almost like your body needs time to cure itself. So it basically takes more of your knowledge today that you have from immunology on the treatment side than actual treatments. But we'll touch base on many of that. So it's basically these two, or these three unconventional agents. Nope. We don't need to go into all of this, but this is going to be important in some cases just because, bacteria especially, they tend to have a slightly different cell, you know, the way the cell is actually formed is a little bit different. But let's go and let's start with the bacteria side. So bacteria, by definition, is a one-cell thing. Okay, so it's a microorganism that, yes, it tends to cluster together and form colonies. Okay, and we tend to basically what we do with bacteria is that we tend to classify the bacteria based in size and shape, in what are the nutritional needs, if it is requires or not oxygen, and basically at the end, if they have an outer membrane or a thick cell wall, and these are the ways in which we classify them. And why we ended up doing that way is because we started to figure out that certain classes of bacteria, they tend to go into similar shapes, sizes, and types of cell wall in actually our membrane, and some other types of bacteria. So we started to slice. And as we moved on into this very traditional Western medicine, what we try to do is like, oh, we have a symptom that is, or attaches that symptom with a specific bacteria or viruses, and that attaches to a specific treatment. Okay, so the idea is that everything would fall exactly as it is, which it does in certain diseases, but it doesn't in many others. Okay, because some of them are not necessarily caused by one little thing or one bacteria or one virus, but that's how we do it. The sizes, and we have, probably you have heard about, you know, bacillus, cocci, or spirochetus, for example, is the name of one. So some of these names, for example, streptococcus or streptococcus or staphylococcus. You know, when you hear these names, in staphylococcus, you probably have heard Staphylococcus aureus is one of those like super bacteria, especially that's very common like in hospital environments. It is freaking, you know, no antibiotics can touch some of those bacteria. And the name Staphylococcus is because you form this like a coccus, like in the case of them is like one little ball, and you bind them together. And as you form like two of them, or a line of them, or like a ball of them, okay, sometimes you get different names. And if you were to look at them into the electron microscope, that's how they would look like. And you're going to see that this is also one of those important things about bacteria, is that they form these clusters, you know, among them. Almost all of them, they form these colonies. Most of the time, you have one technical term that they call biofilm. The biofilm is basically this. The bacteria doesn't occur like in spots, like random, you know, aside in an organ or even in an object and stuff. They tend to really bind together and form like big clusters and big films, you know, in that. And some of them, again, how important that will be for us specifically to solve our case studies. Not too much, okay? But it is important because you hear this all the time. You hear these names, you hear these forms, you hear them all the time. So it is important that you have a little bit more of a knowledge there. These guys here, and you're going to see that, you know, for these guys, and so this is how a bacillus, like the bacillus of Koch, the one that causes, for example, what is the name in English now? The lungs start spitting blood. What is the name in English? I will remember. But it is one famous, and the shape of the bacteria is exactly this kind of like a basket, like it has like this longer shape. Cholera, the one that causes like diffuse or perfuse diarrhea, they tend to have this bended shape, okay? And so all of this, spirochetes, spirulum, so you have some of the other ones that are actually very virulent, you know, cause lots of the disease. They tend to have also this kind of a leptospirosis. They tend leptospira, which is one cause, it causes like very strong like urinary infection. They tend to have more of this twisted kind of shape. So you can kind of identify some of them just based on their appearance on microscope. You won't know exactly which one it is, but it will give you quite a bit of a clarification of those. Again, they all tend to bind together. They tend to bind together. So the first thing that differentiates, for example, these cells from eukaryotic cells, which are our cells, is that they have something called cell wall. Okay, some of them they have very thin cell walls. Some of them they have very thick cell walls. Okay, and the cell wall basically what it does, it provides more like it's almost like you have your membrane, and you have like a mesh, like a net, you know, around that gives you more support, physical support. Okay, Ronaldo, so why is this important for us? First is because it gives rise the presence of a thick or a very thin layer of this cell wall is what gives rise to the classification that is one of them that is the most known for us, which is, you have heard probably of gram-negative bacteria and gram-positive bacteria. It's because of that, because of the cell wall. It's because of how you stain the cell wall. If you have a thick cell wall, you stain in a certain way. If you have very little or almost none cell wall, you will stain in a different way. And then you can already identify. If you look, for example, at gram-negative bacteria, gram-positive bacteria, gram-negative bacteria tends to be bacteria that, gram-positive bacteria tends to be types of bacteria that are more like contagious. It goes, for example, from person to person, or from animal to animal, or from something like an object to an animal. Gram-negative bacteria tends to be more of those like environmental bacteria. E. coli, it's freaking everywhere, you know. So it's around, it can infect you, but it's not necessarily. It's a little bit different in how they infect, you know, a certain host. The reason why they have the cell wall is that to prevent bacteria or helps bacteria or prevent them from rupturing. We understand that these bacteria, they have to live in a certain condition, especially outside the host, and they will survive quite a bit. But if they are in the environment, they are exposed to a lot of changes in temperature, in osmolarity, in presence or availability of water and so on. So they have to basically withstand that gradient, that osmotic gradient pressure. So the cell wall helps them to do that. They maintain the shape and serve as the anchorage for some of the flagella. The flagella is one of those structures that makes this basically the bacteria to swim a little bit. But the flagella is also one of those points that causes antigenicity, you know. So the cell wall itself is one of, or some of the stuff that is inside the cell wall is what's going to cause the inflammatory reaction, you know, from the host. So lots of the antigens that our body will try to fight are actually either attached or is part of the cell wall. Okay. And as I said, lots of, clinical importance here. The virulence factor, which means whatever that is attached or anchored into the cell wall is what's going to give you a stronger or a weaker inflammatory reaction. Super important in the sense of antibiotic action. The very first one to be found, penicillin, for example, acts exactly on this cell wall, on how this cell wall is formed. If you somehow disrupt how the cell wall is formed, all of a sudden the whole bacteria becomes weaker, more prone for rupturing, it kills it. Okay, so that's one of the reasons. And it starts to give you a little bit of this sense that, antibiotics will act on different mechanisms of the survival of the bacteria, which basically means that, not all antibiotics will act on all types of bacteria. So you're going to have some that act in all of them, but you're also going to have a few that are very specific to certain types of bacteria. And obviously they are different from eukaryotic cells. The composition here, like this, one of the most important ones is what we call peptoglycan. As the name sees it, it's a mesh of peptides with glycan, so it's a glycoprotein at the end of the day. So you have some sugars here with polypeptides. This NAG and NAM, these are just names that they give. At the end of the day, you have this very long fiber here that they call that are made of this monosaccharides, and then you have bridges here that actually put those big tubes here together. So you have the monosaccharides and the peptoglycans almost forming like a fabric, a net. That's pretty much what it is. Okay. One of them, or a few of them, so you have some of, the penicillin will act exactly, and is not necessarily on the, I'm not quite sure if it is the NAG or the NAM, but at the end of the day, what happens is that the penicillin will end up acting in this bond here. So by acting on this bond, it doesn't make the fabric a little looser. And by making it looser, the cell wall is weaker. Okay. Very good. So let's go to the first one, so that we call. So on the names, on the shapes, on the forms, on the shape, the size, the colony, it is important that it's not going to have much to do with how we treat, some of the clinical cases that we've seen. This is important. So this is, once you have this gram-positive cell, it basically means that imagine the bacteria will have a few different things inside, of course, but the most different thing is that cell wall. And that cell wall on a gram-positive bacteria is very thick. Okay, so you have several layers of that peptoglycan, layer. So you will have some, and you're going to have a few things attached to this peptoglycan, which we call teichoic acid. So it's a little bit of this, sort of we call lipoteichoic acid. It contains alcohol, phosphates, a little bit of fats or fatty acids. This teichoic acid is one of the major things that causes antigenicity. You have obviously the whole. If you want to make something very specific as what exactly causes antigenicity in most of the gram-positive bacteria, you can call it flagellum, you can call it some of the cilia, some of the glycoproteins around. This teichoic acid is one of them. So lipoteichoic or teichoic acid will cause inflammatory reaction. This is exactly where your immune system, if they have to get a portion of this bacteria to increase the immune response, they will go exactly after this teichoic acid. So this is basically one of the features of the gram-positive bacteria. The gram-negative bacteria, it is a little bit different because it has a very few layers of peptidoglycan, or sometimes just one. So it's almost imperceptible to the staining, and that's why you say gram-negative, because that gram staining doesn't attach too much to the gram-negative bacteria. So it does not contain teichoic acid, but they have something that we call the outer membrane. This outer membrane is, among other things, this outer membrane is basically an extra set of membrane. Okay? So you have the plasma membrane and you have the outer membrane. The only thing is that this outer membrane will have a lot of things actually also anchored on it. You're going to have something attached to it. One of the most important is this lipopolysaccharide, LPS, which many of you already know is causing lots of inflammatory reactions. The same way that lipoteichoic or teichoic acid causes inflammatory reaction on the gram-positive, LPS, or lipopolysaccharide, is the one, or among several of them, this is probably the most famous. The LPS is what causes the inflammatory response on the gram-negative bacteria. Okay. A few things, here you have, a little bit of the kind of a change, or you stratify lipid A, a polysaccharide core, and an O, kind of polysaccharide. Sometimes the O is the one that you see in identification of some of this bacteria. H, N, O. It depends on the bacteria, the strain that you're trying to identify. You have different numbers for this H, N, and O, which basically means depending on how virulent the bacteria is, depending on what is the strength of that bacteria to cause inflammatory reaction in disease, you can have different numbers going there. So if you go and study really microbiology, bacteriology, you're going to have, for us, E. coli. Nice. For those guys, it's E. coli and something, O something, because the strain matters. Okay, that's how much it is important. And when you talk about, the strain, or the strength and the strain of the bacteria, once, I think we mentioned that at some point, that the same way that for the immune system to be effective, and for example, the antibodies to be effective, you have to attach stuff. One cell, they have to physically bind to another. In order for disease or to the colonization of that tissue to happen, or to the immune reaction to happen, this is the same. So a bacteria is more antigenic, or a virus can be more virulent than the other. A bacteria can be more virulent or less virulent, the same way for the virus, depending on how strong it attaches to the cells of the host. So depending on that, and that can vary tremendously. So if you get, for example, the outer membrane, and I'm going to show a few pictures later of the two, but the outer membrane, which is this, this is for a gram-negative bacteria. The outer membrane, the thing that it is sometimes, very is a great feature of this gram-negative bacteria, is that this outer membrane tends to be very, very negatively charged. Okay? It has, glycoproteins in a certain way. And why is that important? Because if it is too negative, it tends to do what? Repel. You repel other cells from attaching to it. So your defense cells tend to have a little bit more of a problem getting in touch with it. Some of the other structures, even the teichoic acids on the cell wall, also tends to have a little bit of that characteristic. So it is just ways in which, you know, either viruses and bacteria, they tend to have to avoid a strong immune reaction. So let's go back to the actual form and the way these bacteria are. So you do have your plasma membrane, similar to what we have, phospholipidic, bilayered, and then you have several layers of the cell wall, okay? And this cell wall, then lots of them formed by this peptoglycan. You have some of the proteins, or sorry, the peptoglycan, which is the NAM and the NAG, followed by the polypeptides and some of the sugars with the polypeptides. And you have some of this lipoteichoic or teichoic acids, you know, kind of going around, and, you know, sometimes they are anchored here, sometimes they're anchored in the plasma membrane, but they tend to cause lots of antigenicity. In the case of gram-negative bacteria, you have your plasma membrane, similar to the gram-positive, but instead of having lots of layers, you have one. And then right after that, you have another sort of plasma membrane, but you call them outer membrane. Okay? Does this make this cell a little bit more resistant to external pressure or osmotic pressures? It does. Okay, so this tends to survive a little more, especially in the outside. But this one here also has this outer membrane. Some of this here, this phospholipid membrane, and then you have this lipopolysaccharide coming out of it, and this is what also causes this. So of all this stuff that we saw now, is like we classify bacteria in different ways. They're important because you can identify them based on certain diseases and also based on their structure. It is important that you know a little bit their structure, because then you can maybe choose the right antibiotic, or you can somehow narrow down what kind of bacteria you're dealing with. For these things, talking about cell wall, it's very important that you understand how it is and what it is, how it is formed, and you know what it does. The outer membrane, also very important, and having the idea that this lipoteichoic acid pretty much works like this lipopolysaccharide in terms of antigenicity of the bacteria. Why do we take, we keep the antigenicity, this? You're going to have in your body lots of these two types of bacteria, especially in your gut, sometimes in your urinary and reproductive tract, in your upper respiratory system. They don't necessarily cause disease all the time. Okay, sometimes you need to have them in a certain balance, and this is probably this stuff much more than when you go and talk about viruses. Especially with bacteria, we understand a lot more today that you have more bacteria cells in your body than actual cells in your body. So they do have a lot of influence on how your body works. They produce stuff that influence how your body works that we still don't know how it does. We just understand that they have a major influence, and perhaps the way we look at today, gram-positive, gram-negative, it has made wonders for medicine, especially Western, but they are, in my opinion, they are still simplistic. But believe it or not, this is still how we do certain things. Gram-positive and gram-negative have been found been there forever. People already know about those things for a long time, but it still is the way in which we kind of deal with, especially on a daily basis of clinical work. That's how you deal. If you want to be very specific and you want to know what kind of bacteria, especially this and that, you go maybe get a sample, do PCR, and you're going to know exactly what kind of bacteria, what type, what strain, and maybe you can be more specific. Still, you wouldn't know if this antibiotic A or B would be more effective. Okay, so one of the reasons why perhaps today we have more and more, especially I think Canada in that sense is better because we have a public health system. The idea of giving antibiotics is becoming like, I'm really going to give antibiotics if you're like 10 days or 7 days at least, you know, suffering from this, you still haven't got better, and this and that, because even for bacterial infections, people are trying to be more like, let your body heal, let your body fight before we enter, because most of the antibiotics we have today that we take 90% of the time, amoxicillin, it's like a bazooka, okay? You have something, boom, you kill, but at the same time, it's too much. You know, you will also affect your GI tract, you also affect other things. So it's the kind of stuff that as we move on, for us, for the class, it's fine because we're gonna try to be very mechanistic, but I think just as we go and look more into the therapeutics of it, we tend to have more and more stuff going. Very good. So, where's the... Okay, so bacteria itself, that's the most important thing we're going to need to know. Those two classifications, certain types, certain shapes, and so on. As we go into the virus will be, like bacteria, we didn't touch base like in detail, but they reproduce outside the cell. They have their own machinery to produce energy, to replicate, to do a lot of things. Virus is really what we call obligatory intracellular parasites. When I say intracellular parasites, is that they can live outside the cell. Of course they can. They can be in a droplet in the air, okay, and still be alive. But in order for replication to occur, is going to be more problematic. The features here is that what they have and one of the things that, you know, and I remember once virus, and the people that studied virus since the beginning, they were, one of the reasons they were very, very impressed with virus is that the virus tend to be the single most, or the simplest of the organisms that can live. Okay, so because they have what is absolutely necessary to survive outside the host, which is like basically some sort of protection to a nucleus, which basically that's what the virus is basically a nucleus, because it just have either RNA or a DNA material that is ready to use someone else's machinery to replicate and to multiply. Okay? So no wonder when we talk about, you know, bugs in our computers, we call them virus. Why? Because it is really like a simple thing that can really derail a very complex machine like a whole computer with all your archives and all the stuff, because it's just it's the simplest of the programs. To look at, you know, viruses cannot, in computers, can never be too complicated, because they have to sneak in stuff and enter your system, and somehow they attach to a very specific thing in your operating system or whatnot, and then causes a disease. Okay, so they tend to be a nucleic acid protein coat. So the protection is just they multiply inside living cells, and they transfer viral nucleic acid to other cells. Obviously, they can be DNA or RNA and a code. So that's basically what it is. In this case, is there anything on identification of this virus? Of course, they have classes of viruses. Okay, so take for example this class of virus that we tend to call herpes virus. It's a class of viruses, and they tend to have these characteristics of latency. It means once you have, it's almost impossible to get rid of, and they have this they're very sensitive, as meaning if your immune system is strong, they'll be very quiet. But if your immune system eventually gets bad, it senses that you have less, inflammatory mediators, less cells around, and they start to multiply. So by the time you start getting, one of the being close to final examination, and all of a sudden, I'm sick. Sometimes it's not necessarily just because you acquired somewhere else. It's just because you get stressed, your immune system goes down, some of this latent virus surface. Okay? This is something, for example, that bacteria, I don't think you have such a thing. You don't have, first because you have bacteria like crazy in your body, but you don't have that kind of, opportunistic bacteria that all of a sudden just senses stuff. No, that occurs in a slightly different way. Very good. So, for the most part, since they have their DNA or RNA material, they tend to have, again, very simple normally it is to replicate, and even the enzymes to replicate, they tend to use the ones from the host, but it's basically protein to produce the coat, and a few enzymes to do actually the DNA or RNA replication. It's an actual very simple DNA or RNA code. Okay? Tend to be very species specific. They can cross, but it's not very common. Especially the coding of the virus, the same way as teichoic acid or LPS and a few other flagellae and cilii from the bacteria are important for the antigenicity. The coating of protein from that virus will be important because you still have to get into the cell, attach, go inside, and promote the disease. So this next slide is nice because it gives you a little bit of an idea of size. So this is a red blood cell. And a red blood cell is not exactly a big cell. Okay? It's an okay size cell, but for example, macrophages, neutrophils are much larger than a red blood cell. An E. coli, so this very, very common bacteria that we find, and this is more or less the size. So everything else, especially these other ones here, are actually viruses. You have this virus here. This is the Ebola virus, which is if you compare to most of the viruses, it is a big virus. Okay? But the other ones are like super small. They tend to be like much smaller than even an E. coli. And if you take, for example, bacteriophages. Bacteriophages are what? They are viruses that infect bacteria. Sometimes they are used even to kill bacteria. And they are like super, super tiny compared to a bacterial cell that in general is very small compared to a regular human cell. Okay? So you're talking about very, very tiny things. Okay? So in those cases, for you to identify, and imagine these guys here. These guys here, not only they are relatively big, but they tend to cluster together and they have forms those biofilms or those colonies. So you can basically go like we have today in microbiology. If you want to identify, for example, a bacteria, yes, you can do the PCR and everything. But because we know what are the conditions in which they live, oxygen, if it's anaerobic or aerobic, what kind of nutrients they like most, you can create what? Specific plates, cell plates, that has the nutrition, and then you incubate in a certain way, that you, if it grows in that plate, it's very likely to be a certain bacteria. Okay, so this is one of the ways, or one of the best ways, the most common ways in which you do identification of bacteria. So in our farm, for example, at the dairy farm, once we have a condition that we call mastitis, and it is infected, so it's an inflammation of the mammary gland. And we know that if we want to treat, and this is one like specific case, if it is gram-negative bacteria, we tend not to treat the cow. If it is a gram-positive bacteria, then we go on and we say, let's wait a little bit, but then we treat. Why? Because we know that for a certain type of bacteria, in that case, some cases of gram-negative bacteria, you have your rate of self-cure is high. So we say, let's not use antibiotics, and let's let the cow treat themselves, with their own immune system. So with the virus, the identification becomes, you can imagine, it becomes a lot more complicated. Okay? Because not only you would need large amounts of these, even to identify on a plate, but in this case, what would be the use of a plate for a virus? Because in bacteria, you can put it on a plate, like a microbiome, like one that can identify Staphylococcus aureus, and you put those, certain things that they like the most, and they cultivate them in low oxygen, blah 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 blah, and all of a sudden they're like, oh, I like this plate. You start eating, you start replicating, you start forming colonies. So the way they identify is that, have you seen those microbiologists? They go, look at the plate, oh, this is Staphylococcus. Oh, this is strep. Oh, this is something else. So they look at the form and what type of plate it is, and they can identify it. With virus, how would you replicate that with something? How do you think, you can do diagnostics of a certain agent, when it comes to virus? Well, it could be PCR. Maybe you can go deep into the genetic coding, and so you can identify some of them. But if you wanna, have you ever seen, like, when you were, sometimes I was like many years ago with upper respiratory infection, and they said, oh, it's probably bacterial. They get a swab, they get a sample, sent to the lab, eventually they identify. But if they want to identify something that is like viral, for the most part, they guess, especially on your day-to-day clinical work, for especially for humans. Because of the types of symptoms and so on, you guess. Bacteria, you do that too, because if it is a upper respiratory infection, or a urinary one, or digestive, or your kidneys and stuff, you know that certain bacteria, have you ever noticed this, that for certain bacteria, you can really try to narrow down to a certain set of symptoms that normally tends to go with a certain set of organs or one organ. Why? Because some of these, either bacteria or viruses, they have a preference for certain tissues. And they sometimes have preference for certain tissues because that specific tissue has something that is pretty good for their growth. Okay? So for example, for example, the rabies virus. The virus that causes rabies tends to travel and go to what? Your neural cells. Sometimes your peripheral nerves. But eventually, if you don't take care of it, it will travel all the way to your central nervous system. But it has a preference for neuron cells. It didn't go elsewhere and stuff. No, it stayed in neural cells. Leptospirosis, eventually, it tends to go to your kidneys. You know, influenza really likes your upper respiratory tract. So the idea is that some of them, they tend to, you're going to see in cases where some of these bacterial viruses, they tend to be very specific, and some others, like fungi. Fungi tends to be more opportunistic, which means fungi tends to be more like, let's say your immune system is down, so they tend to be, the first is like mycosis, like it tends to be like skin infections for some time. Then it goes upper respiratory tract. Why? Because these are spaces or places where you have the most contact with the external environment. So in this case of viruses and bacteria, that's not always the case. Sometimes even if you have more, some of these rabies, it enters, or most of your bacteria or virus infection, sometimes it enters through some lesion, your eyes, or your upper respiratory tract, but doesn't stay there. It really goes in at some point and it finds their niche that they like it. Now, going back to the identification. If you were to do this and identify, saying, I want to know specifically, for bacteria, first line, do a culture them on a plate. For viruses, what do you think it would be? There's no simple answer, but what would be ways that you say, maybe that's a...
Could you try culturing the virus on like a tissue sample?
Yeah, that's it. So what you could do is to cultivate in a cell line, or you could do it even in other animals. You know, you inject and see what happens, and so on. So sometimes for rabies, they you know, I know it was in vet school, you would go into like samples, and you could then inject. Actually, you would try to inject. Today, I don't know if they still do that, but you would inject even inside the brain of little rats. So the idea is that after some days, if the rat became sick, it means that that sample was infected with the rabies virus. But at that point, probably 20 years ago, PCR was not exactly a thing. So today, I think you would have a much better chance of doing that through PCR. But it's still, because of their size, even doing PCR and other things, you would have to have very good samples. Because with the sample, you're going to have lots of other things in the middle. Okay, so you would have to create those protocols, but today I think you do have them. Very good. What time is it? 40. Let's have a 5-minute break. Okay. Do we have any? Oh, they. Good. Okay. No, they're probably gonna have another one. Yeah. Well, let's see.
Okay.
Yeah.
question. In regards to bacteriophages, I heard somewhere that there's potential to use them to treat bacterial infections because they're a lot more specific. Is that tech that's being used?
Yeah. Well, I don't know how much they're being used. I probably not that updated into especially the medical medical on vet sciences. Not yet, but all these things on how do you do these kind of infections and replications? It's something that, for example, using viruses for trying to modify the virus with a certain DNA or RNA that of your interest, and try to make that virus or to enter a cell in order to produce something.
That's pretty cool.
This is already in lab and stuff. In order for you to do, for example, transgenic animals and so on, you have done that. People have done that. But in order to use, a different type of a bacteriophage would be like a vehicle, to a little bomb that that attacks a certain bacteria. It makes all the sense in the world. I just don't know how, like if it is really done today.
Yeah, I think it was maybe like pretty new, and I'm sure things in medicine take forever to get out there.
Yeah. And especially, I can just imagine like the nightmare trying to get something like that approved through FDA. Yes, we're gonna fight bacteria with a virus, and what if that get out of control and da 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 da. But I'm sure, honestly, they will find. People will find a way.
I think they're really specific is why, like, so you can really target the.
Yeah. I think that's the stuff on almost all the everything that I've looked and I see people talking, my colleagues here from UBC and elsewhere, talking about the where therapeutics will go is exactly that. You try to go to something that is less of a bazooka and more of a sniper, and that goes almost all the way from infections in general to especially, like, I think one of the fields that probably will go will be ahead is on cancer treatment, because then what you like the big deal with chemotherapy, radiotherapy is the collateral effects are, I shouldn't say as bad because you can cure a person, but it is really down because you kill them all, including your healthy cells. And they have made amazing strides. The only thing that they have not found yet, and my opinion is that eventually they will find, that when people say that you have, for example, cancer, depends on the stage, what type, yourself, because they say that cancer is really like 5 million types of disease. Some people for the same kind of cancer responds beautifully well to a certain treatment, while other person doesn't. So it's not like antibiotics, penicillin, if it doesn't have like kind of a resistance, it will kill gram-positive bacteria. It's like it's a done deal because it goes into the very core of how the structure of the cell is, fights the very thing. When it comes to cancer treatment, apparently they have like all these receptors and how they replicate and stuff is so different that it's almost like if there is one field where specificity is what they probably try for. You really want to know how specific that is. So the idea of that individualized treatment, or that people in the medical field, they dream of that. That before giving you a certain treatment, they'll go to your DNA, RNA makeup of a certain tissue or yourself, and they will establish that you're more likely to respond to a certain drug, and given that you have this specific bacteria that was identified by PCR, it basically means that, you know, you have this and this drug that will be super specific to you for the disease that you have now. That is the super duper dream of therapeutics today, is that how close, I think we're very far from that. So people like to say they're close. Like the other day, I know we're moving away from the major things, but I love those topics. This stuff is, there is a pretty good or a pretty big topic in the scientific world today, which is, do you remember when stem cell came along like many, many years ago? People said, you're going to basically cure dishonesty, you know, with stem cell research. And you're gonna get, and at the end of the day, I remember those times, like people in research, they were gathering millions of dollars for their labs, you know, to do research on stem cell and this and that. Think about today how many things are actually used therapeutically with stem cell. There are. It is to the hype it was 20 years ago, not even close. But you don't have nearly 5% of what they said it was happening. Why? Because it is complicated. This is one of the biggest things that is happening today in research, is in therapeutics or biology in general, is that, you know, when we talked about these things, the virion, the nucleic acid is a single-stranded or double-stranded, tend to be like a circular DNA, and you have an envelope. This is knowledge from quite a bit ago. People already know these things about the replication of virus, bacteria, gram, like I was talking, gram negative, gram positive. This is like ancient. People know about these things like forever. The problem is, as you move to more complex things from now on, and think about therapeutics. Since you were born, let's say you have anti-inflammatories, you have antibiotics, you have different forms of how you can inject, you can, is topic, is this, is that. But think about the amount of weapons you have today compared to when you were born and now. Do you think it has changed like tremendously? It hasn't. I think what has changed in a few fields like cancer, the treatment from 20 years ago are completely different. They are so much better now. They're like some specific things for HIV/AIDS. 20 years ago, it was a death sentence. Today, you have Magic Johnson. The guy is like 20-something years, 30 years, I think, with HIV. He's a billionaire businessman doing all kinds of stuff, you know, So he has changed many things, but on your day-to-day things, yeah, you have better antibiotics, you have maybe some more potent things here and there, but it's not like he has changed. Like most of the stuff that increased your quality of life and your longevity has to do with your lifestyle from 20 years ago. From 100 years ago, I agree. Antibiotics and all the stuff was clean water for the most part. But now, for the 20 years from now, it hasn't. So it's the same as microbiome. We're talking like one of the hot topics today is microbiome. So you have, the microbiome of an obese person is different. So if I inject the microbiome of a thinner person into an obese person, this obese person will turn into a thin person. That's the whole thing. But now they're looking at whatever your gut produces affects your mood, affects your immune response, it affects stuff. And for you now to dissect those things is going to be complicated. It's going to be complicated because our system is made for what? You write a proposal, you write something, or a company, or a government, or a researcher, or whatever, and you try to solve one slice of the problem. And by solving the slice of the problem, you say, okay, this is the direction. And then somebody else is kind of finding out something completely away from that, and it found exactly the opposite response because the conditions were a little bit different. So right now you would have to have almost every project for these bigger questions will have to be big projects that is not necessarily led by one person, but they have to be connected to each other. So it's more like, it's not you creating one new crazy thing. Obviously you still have that, but you're gonna have to combine a bunch of things. And we're still not very good at combining a lot of things. So that's my long response. That's good. So let's go. So we talk about the viral structure. So the same way as we were having like the plasma membrane, cell wall, outer membrane, teichoic acid, LPS. For the virus is the virion. The virion is basically a complete package of a virus. It's the proteic protection, the capsomer, or we call the capsid, and with the DNA or RNA inside. That's it. Virus is like, wow, cannot be simpler than that. Okay? And probably at this point, we haven't found anything that is simpler than a virus. Okay? We haven't found a being, when I say a being, like something with a genetic material that can have. That's why prions was that kind of, whoever found what prions was even won the Nobel Prize, because prions is that weird. It's this thing, it's like a protein that all of a sudden turns into a weird protein that affects whatever. So it's it's different than what we always consider to be life. Life to us has to have a genetic material, some form of coding there, okay? Which prions is like, yeah. So the virus structure, I'm gonna say the next slide will show better, but you have the capsid and this envelope. So capsomers is basically a bunch of little balls that goes together that forms the capsid. The envelopes, sometimes not all the viruses have the envelope, but it is basically, and sometimes the envelope can be, what parts of the host cell, because once it removes from the cell, sometimes it can be enveloped in a little bit of a layer of the cell itself. And you have sometimes these spikes here, which we use for identification. So that's basically what it is. You have the capsid, which is this whole blue thing, which are formed by capsomeres. The nucleic acid is arranged inside. It is not arranged necessarily the way it is in our cells, but regardless, still DNA or RNA material. Sometimes you have the envelope. So this would be the capsid of this specific virus. Sometimes you have another layer outside, and sometimes you have some of these spikes. These spikes, they are used sometimes for agglutination between viruses, but also as attachment points to the host cell. Okay, so they can be used by that. Sometimes, they can be used. This is an electron micrography, and what you have is like sometimes you have these dots here. They say, oh, this virus has a spike, so it's much likely to be this kind of virus here and there. So used for identification, not to the same extent that we use staining for bacteria, but it can be eventually used for identification. But this is the baby. Okay, this is the little thing that can cause you a lot of trouble. Cannot be simpler than that. Okay. For the most part, is the virion structure. So basically, the capsids and a few enzymes. That's what the RNA and DNA are for on this virus. Even all the enzymes that are needed for the replication is not in their DNA RNA. They basically use whatever's in the cell and replicate. Okay, so intracellular activity. Sometimes you're gonna have like lots of them in a single host cell. As I said before, certain types of viruses, they have the ability to sense if the immune system is strong or not, and just basically stop replication. They just stay in that cell, and they stay at such low levels that even natural killer cells and some cells that go and try to, get samples of every cell to see if everything's okay, that cell would pass, as normal, because they're basically quiet and not replicating. Okay. Again, this is something that is very different from bacteria. Bacteria, oh, there's a little bit of blood somehow, but it replicates. Okay. It doesn't have that control. Consequence in host cells, for the most part, once you have thousands of virions, in the same cell, what probably will happen is that the cell loses the ability to, because the virus takes control of all that machinery to produce other things. You end up weakening the cell, and eventually you produce so many of this virus that the cell explodes, basically explodes, and then you kill the cell. What they try to do is basically to you have some receptors, or you have some sites that can attach the virus to the plasma membrane. The spikes would be probably one of those examples of things. It's not the only thing that attaches a virus to a host cell, but it's probably an example. You're going to have some sort of that protein itself that forms the capsule actually forms some sort of receptor, and with that it attaches to the cell, and from there it can basically go into what they call, they form little vesicles that basically protects the virus, as it enters the cell, and eventually, this cell, and this is the stuff that I'm not entirely sure how is the direction once the virus enters the cell. I know, from my knowledge, is that he has a way to go straight to the nucleus, and from there somehow incorporate into the DNA of the host cell.
When your immune system is trying to differentiate between the cells and non-cellular cells, and do they detect the attachment of your virus, and that's how they determine if it's a cell?
No, I think he has to do more with the. I think the attachment at the end of the day is not going to be enough to. But if you have a cell, and this is a normal cell, and this is a cell that is infected with a virus, basically your MHC1 eventually gets a sample of this and shows here. Okay, and here is going to be the same. The only thing is that this one here will have some viruses around. And when I say viruses around, it's not necessarily the virus itself, but your intracellular ecosystem is different. Because imagine that all of a sudden your cell stops producing a lot of things that makes that cell healthy. And all of a sudden it starts producing only virus, period. So it means that the whole makeup of your intracellular media is different. So that's where it goes and say, oh, this thing here, and eventually, yes, there are viruses there. Okay, so you do have lots of viruses. So that's how it catches. So from what I understand, you're not going to be able to, unless I think the cell is too sick, you're not going to be able to go and say, oh, this is, it is based on something that is different here or here. But even in cells that were, that have been cancer cells or whatever, you would have to depend on what's inside, the cytoplasm, not necessarily the membrane. Very good. Listen, all the other ones here, we can go into more details on this. Let's see. Yeah, let's just stop here, and then we go into the actual multiplication of the viruses. It's not in maybe 20 minutes in the next class we'll be done with this part. But it's basically saying that for some of them they will have to go inside, they will have to replicate, they will have to coat, you have to produce the coating, and then you have lots of viruses going around. One of the other things that is also important, and this is much more for viruses than for bacteria, and the reason why there's this immense concern with how contagious the virus is that because of their size, you have a droplet of that you're like, you cough, or you have you can have like millions of those. And that's why, the idea of covering or doing something is so important, because much more than, can you do that, it's, because of contact with bacteria, yes, you can. But viruses tends to be, because of their size, they tend to be like, yes, a bad cough in your face, yeah, it's enough to pass, the disease to you. Okay. What we're going to try to do is finalize this class, in the next class, on Thursday. We're probably going to get one of the other case studies also to start, because one thing that is happening now is that even for the pharmacological interventions, it's some of the stuff, first of all, you have some knowledge of it. Not necessarily that we're giving crazy new things here. But at the same time, is this kind of approach to, when do I give and why, and how this can affect your immune system and how your immune system. Now we're going to go and try to go into the complete case scenarios, because now we have more or less like a pretty good chunk of homeostasis, immune system, blah, blah, blah. And now we can have conversations about that. And this is exactly what I would like to do also with your presentation. As much as the presentation, yeah, it's a little stressful, you come and present. The idea is that even for the, when you present for the others, is for you to try to imagine things. The class is less about the right therapeutics, and more about you coming, what if I do this? What if I do that? What if for a case of viruses I inject something else that has nothing to do with treating the virus? So that's the whole point. So it's supposed to be more fun now. You can do whatever. There's no wrong answer, but that's the idea. So we'll continue with this, finalize another case study on Thursday, and then next week, or at least the idea, maybe we can start pharmacological interventions on Thursday, and then we finalize next week. Okay? Very good.